Hello, everyone. This is Steven again, and again, these lectures are not lectures. I guess these are more just review sessions for my microbiology test coming up on tomorrow actually. And so I'm going over all the slides that were produced by my college, Sinclair Community College, in an effort to review the material and help me review it and maybe help others learn it potentially. So that's why I'm doing these. So I hope you're having a good Memorial Day. It's Memorial Day here. And today we're going to be talking about eukaryotic cells. This is chapter 5 in the Microbiology Cowan textbook. And so let's get started. This is a short little slideshow, so it won't take too long. If I can get this to move. There we go. All right, so here we have a picture of an overall eukaryotic cell. This is going to be a plant cell because we have a chloroplast here, organelle down here. And as you can see, all the different organelles and all the different aspects of the cell are labeled. We have the rough endoplasmic reticulum that is coming off the nucleus. So the way to talk about this is to sort of think about it from the perspective of creating proteins. So inside of our nucleus, we have our nucleolus, which is where a lot of the DNA and other nucleic acids are synthesized. And what that does is it goes out through these porins here in the nuclear membrane of the nucleus into these rough endoplasmic reticulum. And these rough endoplasmic reticulum, or RER, have these ribosomes on them. And the ribosomes are where the proteins are made. However, these ribosomes can also be free out in the cytoplasm out here. And so what happens after these proteins are synthesized, they get further modified and packaged in the Golgi apparatus, which is right here. So you can think of this as sort of the factory here. And then this is sort of the distribution center for the proteins of the cell. Let's see, other important aspects of the cell. We have the mitochondria, where the energy is made, where ATP is made. In a plant cell, we have a chloroplast here. That's how the cell makes its own energy from photosynthesis. We have these centrioles. We have a glycocalyx, which is a outer area, which is typically more characteristic of a prokaryotic cell, but I suppose they're trying to tell us that these can be on eukaryotic cells as well. The lysosome is where things are broken down. Let's see here. Microtubules are important for transport across the cell. So these are sort of like the cellular highways. We have the microfilaments, which help keep the cell's structure, as in the bacteria cells. And let's see, if it's a plant cell like this is, we have a cell wall. So as you can see, the footnotes, a structure not present in all cell types. Same thing with a flagellum and the glycocalyx and microvilli. Okay. So here's some of the things we're going to be going over today with eukaryotes. Some of the historical and evolutionary perspectives, the form and function and the different structures on the external as well as the internal side of the cell, and some of the pathogenic eukaryotes. So I mentioned in a previous slide overview that it's believed that eukaryotes came about because larger prokaryotes inhaled other eukaryotes. Larger prokaryotes inhaled other prokaryotes, and they sort of inherited their function. So as you can see here, that you have an ancestral prokaryotic cell, and what it does is it's enfolding right here, and it's absorbing another prokaryote. So we're inheriting the function of this prokaryote, and then we get another prokaryote from this plant cell because we have a photosynthetic prokaryote, which is going to be those chloroplasts. And so this is sort of how those mitochondria and chloroplasts are believed to have originated in eukaryotic cells, is because they were absorbed by other prokaryotic cells that happen to have a nucleus, like so. And sort of this endosymbiotic theory. And here's another breakdown of that aspect. larger prokaryotic cell and a smaller prokaryotic cell coming together. Excuse me, I just ate, so I'm trying to speak clearly here. So we have a larger prokaryotic cell and a smaller prokaryotic cell coming together and forming one higher functioning cell. And there's some different evidence for this in the fact that the mitochondria and the chloroplast have 70S ribosomes. They divide independently within the cell, which is interesting. They have a membrane structure. So if you remember, those mitochondria have that double sort of inner membrane and outer membrane by themselves. So that's sort of one reason why they're believed to have lived independently at one point. They resemble a cell membrane of gram-negative bacteria, and the chloroplast resemble a structure of cyanobacteria. The form and function, some of the external structures of a eukaryotic cell, we have flagella on some types of cell, as well as cilia, cell wall, and a glycocalyx. And here's some pictures of that. A flagellum here, and some cilia. So these flagella and cilia are made of microtubules, which, as you can remember from that other slide, they're also on the inside of the cell as well. They're important for motion. They're made up of this protein called tubulin, and from what our teacher told us, that they're famous for these 9 pairs plus 2 arrangements, so it's like a 9 plus 2 setup. So I think that's based on the fact that we have 1, 2, 3, 4, 5, 6, 7, 8, 9 of these doublet microtubules, and then we have 2 central microtubules on the inside of those flagella and cilia. They're used for locomotion. Flagella are long and whip-like, and cilia are shorter and more numerous on the cell structure. The cell wall. The cell wall are present on eukaryotes in plants, algae, and fungi. They're made of carbohydrates, which are chitin in fungi. You get cellulose in the plants, the cellulose pectin, and silicon dioxide in algae. So you get those different structures there. And I believe silicon dioxide is a component of glass, which is another interesting fact. The glycocalyx, which from our previous video was also on the bacteria making up that biofilm, if you remember correctly. The carbohydrates are extending from the animal plasma membrane, and they're present on some cells that lack a cell wall. So they're same sort of functions as in bacteria, where they function in protection, adherence, and cell signaling. The cytoplasmic membrane on the eukaryotic cells, similar to a prokaryotic membrane, where we talk about that fluid mosaic model of the proteins and phospholipids. So you can see the phospholipids here, their phosphate heads and their hydrophobic fatty acid tails. And you get the glycolipids embedded in the membrane at different points, as well as some different peripheral and integral proteins. So we have this guy, and then we have cholesterol as well, those sterols, which help maintain the structure of the cellular membrane. And actually, important to note, this is unique to eukaryotes, the sterols add rigidity. Greater variety of phospholipids and carbohydrates in that cellular membrane here. But the membrane is functionally less versatile than a prokaryotic membrane. The organelles in the eukaryotic cell are all membrane bound, so that's a difference from the prokaryotic cells. So these are some of the different organelles and what they contain. We got a nucleus, which contains the chromosomes, which help create our proteins. We got the endoplasmic reticulum, which is for a transport network, ribosomes as a protein factory, Golgi complex, membrane formation and secretion, lysosome, digestive enzymes, and so on and so forth. Cytoskeleton, that's for those microfilaments we were talking about earlier. So the nucleus, we can see a picture of that here. We got our nuclear envelope that's surrounding the nucleus. We have our nucleolus, which is sort of the integral portion of that nucleus where the proteins, or where the DNA is made, I believe. We have that chromatin within the cell as well. some of the nuclear pores, nucleolus, nuclear pores. And as you can see, we have the endoplasmic reticulum that's sitting right outside of that nucleus to basically take that mRNA and make it into proteins. Endoplasmic reticulum. So we have that's right outside the nucleus, as we said earlier. The ER has these folds called cisterns, and the rough ER has the ribosomes on them, so it's kind of studded like this, like so. And the smooth ER, which doesn't have ribosomes, is nice and smooth. The ribosomes are different sizes. So ADS, the S stands for like a sediment. I believe it's a sedimentation factor, which is based on how it separates in a centrifuge. So you don't really need to know exactly what that means. All you need to know is that ADS is a bigger protein than 70S, or it's more dense, I believe. Membrane bound, attached to the ER. These are the ribosomes that are actually on the rough ER, and there's also some free ribosomes in the cytoplasm. And in the mitochondria and chloroplasts, we have these 70S ribosomes. The Golgi complex, which is outside of the endoplasmic reticulum, which is the purpose of this is to transfer and modify those proteins that are made in the ER. They contain secretory vessels, transfer vesicles. The folds are also called cisterns, as you can see here. And you have this transport vesicles coming from the rough ER to modify that protein even more. So again, with nature's assembly line, here we have the nucleus, these nuclear pores here, nuclear envelope, which is the sort of the nuclear membrane, goes to the endoplasmic reticulum, and then at the sub level of that, you have these ribosomes, which have these two subunits here, where the mRNA is coming through, and these protein chains are starting to be formed here. Another depiction of that. Ribosome portions are coming from the nucleolus, through the nucleus, through these transitional vesicles from the rough ER, down to the Golgi apparatus, into these condensing vesicles, and secretion by exocytosis. The lysosomes originate from the Golgi apparatus, and they contain enzymes involved in intracellular digestion. So if you can see here, a food particle comes into the cell, and it gets absorbed into a membrane here, and then it makes a little food vacuole where the lysosome from the Golgi apparatus, comes up here to the food vacuole, combines with it, and then digests it, like so. So the lysosomes originate from the Golgi apparatus, like here, and they do their thing, they digest. Mitochondria are the powerhouses of the cell. They're the organelles that actually make our ATP, make our energy. As you can see here, there's an inner membrane up here, inner membrane and outer membrane. These cristae are the folds that make up that inner membrane. We got these ribosomes on the inside of the mitochondria as well, these 70S ribosomes. Chloroplasts, which are only present in plant cells, are made up of these stacks of what they call thylakoids. The individual stacks itself are called granum. The thylakoids are the multiple stacks. It's surrounded by a double membrane as well, like so. There's a real life picture of it. The cytoskeleton is what gives the cell its structure. So we have these microfilaments, which are thin protein strands that attach to the cytoplasmic membrane and form a network across the cell. These are mainly important for structure. And the microtubules, which are thicker and more hollow, help maintain cell shape and transport substances across the cell. And we have a little depiction of that down here. The microtubules look like these big old tree branches, and then the microfilaments are these smaller ones. And I think there's actually a middle size one as well. There's a intermediate filament, which is not indicated here, which is probably not important for this class, but just so you know. Some pathogenic eukaryotes, which is the last part of this little presentation here. These are some fungal infections for humans. They can be superficial, such as things like jock itch, outer epidermis, Tinea versicolor, candidiasis, which is another fungal infection, yeast infections caused by these different fungi over here. Systemic, which are more on a deep level and which affect us more. They can affect the lungs or the skin, such as those listed there. Some other pathogenic eukaryotes, like amoebas, which can live in water, and then flagellated protozoa, and so forth. Some flatworms and roundworms, which are also part of those eukaryotes. And that's it. Hope you enjoyed. If you have any questions, please leave them below. And if I missed anything, please comment that as well. Thanks, and hope you have a good rest of your day.